Hi, welcome back to lecture number 3 in our central nervous system unit, the CNS. In our last lecture, we were going over the cerebrum and the cerebral cortex. And this one, we're going to finish it off, and we're also going to go into some subcortical or subcerebral structures. So these are structures that are located deep within the brain, deep to the cerebrum. All right, so if you remember the primary somatosensory cortex, that's where all the sensations in your body, that's the part of the brain, the cerebrum, it ends up in that region of the brain. This question is asking, what's the relationship between the size of the cortical, cerebral cortex area and the sensations from the body parts? The answer is the size of the cortical area is devoted to a body part depends on how many sensory receptors are on that body part. So body parts that have a lot of sensory receptors include your mouth and your tongue and definitely your hands. So we're going back up here. So remember, here's your primary somatosensory cortex right here, located on the post-central gyrus. A gyrus is a bump. So this is a bump. It's post-central because it's located posterior to the central sulcus. It's right here. But anyways, so difference like your mouth and your lips and your tongue are going to have a large area of the primary somatosensory cortex devoted to it. Same with your hands. I don't know if it's exactly right here. I'm just making it up. So your hands also going to have a large area of the primary somatosensory cortex because they have tons of sensory receptors because those are the parts of your body that you're doing most of your feeling with. You're not feeling stuff with your elbow. If you want to see if something's hot or if something's soft, you don't touch it with your elbow or your bicep. You're going to touch it with your hands. So things like your arms are going to have a very small little area of the primary somatosensory cortex devoted to that. And here we have what's called the sensory homunculus. Now, his body parts are weird size because it's representing how much area of the primary somatosensory cortex is devoted to these different regions of the body. So you can see he's got these giant hands. Let me switch the color. Big hands, big lips, tongue. So that's showing that those regions of the body have a larger area of the somatosensory cortex devoted to it. Now, in my class, we're doing the 2-point touch discrimination lab. So where we have to the students have to see if are they getting poked with one pin or are they getting poked with two pins, and they're testing different spots of their body. So their hands are very good at noticing if it's getting touched with one pin or two pins that are really close together because there's a lot of sensory receptors on your hands and your fingers, also your lips. And then they're doing other parts of the body like the back of your forearm that has very few sensory receptors because you rarely use your forearm or your elbow to feel something if it's warm or if it's soft or if it's hard. But you do use your hands quite a bit. So these are areas of your body that have a large area of your somatosensory cortex devoted to it. And very similar to the sensory homunculus, here's the motor homunculus. So this guy represents how much part of your primary motor cortex is devoted to different regions of the body. Remember, the primary motor cortex is located on the precentral gyrus, just anterior or in front of the central sulcus. So once again, giant hands. Giant hands because you need very precise movement with your hands, whether you're typing or you're texting or you're doing sign language or whatever. You need very precise movement with your hands. You don't really need precise movements with your arm. I mean, even if you're throwing a football or a baseball, I mean, that is a complicated movement, but it doesn't have to be as precise as things with like your hand, and especially like your mouth and lips and your tongue. Just forming sentences takes very precise movements. So these large areas on this guy, those have bigger areas of the primary motor cortex devoted to them. And basically the same thing I said on the last slide. So what is the relationship between the size of the cortical area, primary motor cortex, and the type of movement? Well, it's the same thing. The more precise movements that are required, like if you want to spell your name or maybe draw a nice picture, it takes a lot of precise movement of a pen with your fingers to get what you want to spell or what you want to draw. So more areas devoted in the primary motor cortex to the things like thumb, fingers, and your tongue than say your trunk. Because although you do movements with your trunk, you do twisting or maybe sit-ups, those don't have to be precise. But with your hands and mouth, those movements have to be very precise. And now cerebral white matter. Remember, white matter looks white because it's made of mostly myelinated axons. The myelin looks white because it's made of mostly fats or lipids. Remember, on the most superficial region of the brain out here, the cerebral cortex, that's gray matter. It looks gray because it's made of mostly neuron cell bodies. And then deep to the gray matter of the cerebral cortex, you'll start to get the white matter. And now, remember, white matter inside the central nervous system, those are called tracks. Now, once the white matter, the myelinated axons, exits the central nervous system into the peripheral nervous system, or PNS, then they're called nerves. I know in our review packet there was a question that was something like, what are the three jobs of tracks inside the central nervous system? Well, here they are right here. Number one, association fibers. These are tracks that connect different areas of the brain in the same cerebral hemisphere. So, say, like in the left cerebral hemisphere, if some white matter, if tracks connect two different parts in the same side of the brain, those are called association. Another one's called commissural, commissures. And the main one you're going to need to know here is called the corpus callosum. And these are white matter. These are tracks that connect areas in opposite. So this is the corpus callosum is what connects the left and right cerebral hemispheres. So you'll definitely going to need to know the corpus callosum. And the third function of tracks or white matter in the CNS is called projection. Projection fibers. So these are the ones that are going to take the information, the nerve impulses from the brain. It's going to go down into the spinal cord, and eventually it'll reach into the peripheral nervous system. So commissures connect the left and right cerebral hemispheres. Association fibers connect different parts of the brain in the same cerebral hemisphere. And projecting fibers are going to take it from the brain and then project it down through the spinal cord and then eventually through the rest of the body. Here we have what's called the basal nuclei, also called sometimes, you'll hear it just as often called the basal ganglia. But remember, nuclei is more accurate because a cluster of neuron cell bodies, which makes gray matter because cell bodies look gray. So a cluster of neuron cell bodies inside the central nervous system, the brain and spinal cord, those are called nuclei. And if it's a cluster of gray matter, neuron cell bodies outside of the brain and spinal cord in the PNS, those are called ganglion. All right, so this is the basal nuclei. All right, so here you can see the basal nuclei or basal ganglia here in green. Now, don't worry about what makes it up. Like the amygdala, that's limbic system, But so this is located just lateral on each side of the thalamus. You can see the thalamus here in orange. All right, so these are cluster of neuron cell bodies located just lateral to the thalamus, made of mostly that you're not going to need to know what it's made of, so don't worry about it. You need to know what it looks like and what it does. But it's made of what this is called the caudate nucleus. The lentiform nucleus is made up of the putamen and the globus pallidus. So you're just going to have to remember what this shape looks like of the basal nuclei or basal ganglia. It's going to look a lot very similar to what's called the limbic system. And they're both going to be green as well. So you're just going to have to kind of know what it looks like and also what it does. And what it does is it regulates voluntary motor movements by modifying instructions sent to skeletal muscles by primary motor cortex. So basically, it doesn't really, and it says it initiates and it definitely terminates large movements. It doesn't really initiate them, it allows them to be initiated. So what it does is it connects here with the primary motor cortex, which is located just anterior or in front of the central sulcus right here. Here would be the primary somatosensory cortex. And it connects with the motor cortex, and it plays an inhibitory role. So it inhibits those neurons from firing. It either hyperpolarizes them, so it inhibits them from firing action potentials. And if your basal nuclei is damaged and it's no longer inhibiting your primary motor cortex, that can cause just random spasms in your body. So it doesn't really initiate the movements, but then it allows those movements to be initiated by stopping. It stops inhibiting the primary motor cortex. And some other functions of the basal nuclei. It assists in automatic movements like swinging your arms. So when you're walking, nobody thinks about, hey, I got to swing my arms. I mean, it just happens totally natural. That's because of the basal nuclei. So just know what it looks like. Know that it allows muscle movements to be initiated and terminated. And if it gets damaged, it can cause tremors or spasms in the body. All right, so what can happen if your basal ganglia gets damaged? One thing's called Huntington's disease. It's very rare. It's inherited, so you're going to get it from one of your parents. It's autosomal, which means it's on the autosomes. The autosomes are chromosome pairs number 1 through 22, which are the same whether you're a boy or a girl. Chromosome pair number 23, those are the sex chromosomes, where if you're a girl, you have two X chromosomes. And if you're a guy, you have an X and a Y. I can see right here it's on chromosome pair number 4. I did not know that, so you definitely won't need to know that either. It's also what's called a dominant trait. So if you have two alleles for each trait, you get one from mom, one from dad. If something is recessive, like blue eyes, to have blue eyes, you got to have a recessive allele on both of your chromosomes, the one you got from your mom and the one you got from your dad. Dominant traits, on the other hand, you only need one. If you have one dominant allele from your mom or from your dad, you're going to have this disease. But this also makes it's kind of good that it's dominant, because you would know if this disease is in your family. Because if you have one dominant allele for this disease, you have the disease. So you don't really have to worry about this, because you would know if your family carries this or not. So this is caused by a mutated gene, and it causes the neurons in your basal ganglia nuclei, to start to degenerate, to break down, to waste away. And they can see it if they take a MRI or a CT scan. These are the lateral ventricles, large spaces between or inside the brain that holds cerebrospinal fluid. And people with Huntington's disease, those lateral ventricles start to expand and get larger. So who it happens, usually the symptoms start showing up in their 30s or 40s. And what are the symptoms? Remember, the basal ganglia, basal nuclei, plays an inhibitory role to the primary motor cortex. So when this starts getting damaged, it stops inhibiting the primary motor cortex. And then you can get that involuntary jerking movements. Involuntary jerking movements of your arms, basically any parts of your body, your eyes, impaired posture, balance and coordination, difficulty with speech and swallowing, and some other things: cognitive difficulty focusing, stuck on a thought. Psychiatric, you have insomnia, have trouble sleeping, social withdrawal, fatigue. But I mean, if you're fatigued and you have difficulty focusing, don't be all scared that you have Huntington's disease, because you would know already, because one of your parents would have it. And just a little more on Huntington's disease. It gets worse over time. It's progressive. Eventually, the people will be basically wheelchair bound, and they won't be able to take care of themselves. And from symptoms to death, it's about 10 to 30 years. So it is eventually fatal. And remember, this is caused by atrophy, or the wasting away, the breaking down of neurons inside the basal ganglia. It says ganglia here, but remember, basal nuclei is a better term for that. And very kind of similar to Huntington's disease, you have Parkinson's disease. Probably heard of this one more. It's more common than Huntington's disease. This is also a progressive disorder of the basal nuclei. It says ganglia, but basal nuclei or ganglia. And it's specifically a group of structures called the substantia nigra. You can see it right here, located just below the thalamus, right there. And this is what these produce: a neurotransmitter called dopamine. All right, so yeah, the cells in the substantia nigra produce dopamine, and it's dopamine neurotransmitter as well as acetylcholine that are the two primary neurotransmitters that do all your skeletal movements. So the symptoms are going to be kind of similar to Huntington's disease. You're going to have tremors, but it's not going to be quite as jerky as Huntington's disease. It's more of a rhythmic kind of involuntary shaking of muscles. And some other signs and symptoms of Parkinson's disease, other than the rhythmic little shaking or tremors in your hands and your limbs. Bradykinesia, that's just slowed movement. Rigid muscle, impaired posture. Speech can be disruptive. Difficulty writing, of course. If your hand is shaking, it's going to be difficulty to write. Swallowing and chewing problems. Cognitively, difficulty thinking, depression, and your sense of smell might be dysfunctional. And here we have the limbic system. So remember, I told you earlier, so this is the basal ganglia right here. I inserted this little picture of the basal ganglia. Remember, made of the caudate nucleus, the putamen, the globus pallidus. Remember, you will not need to know those. You won't need to know caudate nucleus and that. You just need to know what the basal ganglia, basal nuclei look like and their jobs. And now here, so it looks very similar. Here you have the limbic system, but you can see how the basal ganglia or basal nuclei is much thicker, where the limbic system, it's much thinner. They're both green. They're both located right next to each other, but you're gonna have to know the difference, just by looking at the picture, the difference between the basal nuclei and the limbic system. Now, unlike the basal nuclei, I do believe you're going to need to know what makes up the limbic system. And there's really only two things you're going to need to know. Right here, number one, the hippocampus, right here. Hippocampus is involved in storing new long-term memories, what's called episodic or declarative memories. So these are memories like, hey, do you remember your 16th birthday? Do you remember that? Those are episodic or declarative memories. So it's not like remembering how to ride a bike. That's like a procedural memory. So these are memories that you can specifically recall. So the hippocampus is important in that. And then you're also going to need to know right here the amygdala. So the amygdala is part of the limbic system that's for mostly emotions, rage, anger, stuff like that. I think we're going to do. And then also the olfactory bulb, that's for smell. But the two main things you're going to need to know about the limbic system: what does it look like? What does it do? And also know it's made up of mostly the hippocampus and the amygdala right here. Also the olfactory bulb, but the two big ones, amygdala and hippocampus. All right, now the amygdala right here, part of the limbic system. So they're a pair of almond-shaped structures in the frontal portion of the temporal lobe. To see them, you have to do a sagittal plane, not a mid-sagittal, because the amygdala, the limbic system, is just lateral to the midline, just lateral to the thalamus. All right, so what does the amygdala do? It's like the instinctual fear and anxiety center of the brain. So it makes you aware of your surroundings, and like maybe sometimes you're like, whoa, something just doesn't feel right. So that's like the amygdala. Also things like rage and anger, amygdala. It's helpful in storing highly emotional memories. So if I don't know, the death of a family member, maybe the birth of a child or something that's really highly emotional, it helps to store those declarative episodic memories into long-term memory. And the other structure, cluster of neuron cell bodies, gray matter inside the limbic system that you'll need to know is called the hippocampus right here, called the seahorse of the limbic system because it's supposed to look like a seahorse. I don't really see that too much. Its function is to help store long-term memories. So these are declaratives or episodic memories. So these are not like remembering how to, type or play a piano. These are things that you can actually recall. Like maybe you won a soccer game or something. Those are declarative memories. Now, people with damage to their hippocampus, they get what's called anterograde amnesia. Now, retrograde amnesia means you forget things that have happened. So you can't remember maybe your name or where you live or anything. That's retrograde. Anterograde amnesia is where you remember everything from the past, but once your hippocampus gets damaged, you cannot store any new long term memories. So I don't know if you ever seen the movie 50 First Dates, but she had, I don't remember who starred in it, but she had damage to her hippocampus, and she remembers who she was. She remembers everything from her past, but every day she woke up, it was like she didn't know anything that she had happened the day before. So she met a guy and they started dating, but every single day that she woke up, it's like she met him again for the first time. So the hippocampus is important in storing new long-term memories into the brain. All right, so that's it for lecture number 3. Looks like about 18, a little over 18 minutes long. In lecture 4, I'll be back to talk about what's called the diencephalon, the thalamus, hypothalamus, epithalamus, and also go over the 3 parts of the brainstem: the midbrain, the pons, and the medulla oblongata, and maybe the cerebellum. I'm not sure. All right, so I hope you enjoyed it, and I'll see you next time.